Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta from IIT Delhi. Today we are going to talk on the module Avidity, the nature of antigen-antibody interactions. And of course, this also belongs to the paper called Immunology. Now, much of our insight about the interactions between antigen and antibody has come from simple systems. That's how these studies were carried out by Landsteiner. So in that sense, the discovery of the concept of the hapten was very valuable for Landsteiner in those studies, because having the possibility of generating an antibody which is specific to a small molecule means that it is very easy to understand the structure and activity correlation with the help of making small changes or large amount of changes in these small molecules called haptens. So much of our understanding about this interaction, or more of the specificity of these interactions, that has come by looking at how the haptens interact with Fab fragment, because it is Fab fragment which constitute the binding site. The other part of the antibody, as we have already seen, is biologically not irrelevant. But if we want to understand the nature of this interaction of an antigen molecule and the binding site on the antibodies, Fab fragments constitute self-sufficient, good enough small fragments. Choosing these small molecules gives another advantage, because many techniques, in particularly X-ray crystallography, can be used for this. It is very difficult, much more difficult to crystallize two macromolecules together. However, if you have a small molecule like hapten, and on the other hand you have a reasonably good sized polypeptide fragment like Fab, then it becomes easier to co-crystallize them together and look at the details of the fine structure of this complex. And that is how X-ray crystallography, like in many other areas of protein structure and protein-protein interactions, here also this technique has contributed some valuable insight, particularly at the early phases of these studies in the area of immunology. So let us define broadly the objectives of this module. One, of course, is to understand the nature of binding site on just Fab fragment in more detail. We will also try to understand the origin of multi-specificity. That's a phenomena which tells us that this specificity between the antibodies and antigens is not absolute. Many times this confuses the issues, because all the time while describing various biological interactions, we tend to emphasize the specific nature of those interactions. However, biological specificity is never absolute. And right in the beginning, and that is something which we sort of tend to forget, that if this specificity was absolute, Jenner would never have succeeded in vaccinating people against smallpox by using a cowpox and derived antigen. So we'll try to understand a little bit more sort of a contours of this multi-specificity, how does it arise. That of course is a part of the overall story where we will try to understand forces which govern the binding between antigen and antibody. Because at the end of the day, it is these forces which define the specificity of any interaction between two proteins. In antigen antibody interactions particularly, the concept of avidity is very important. We will see that how does it actually differ from a concept of a simple binding constant. We will also look at the techniques like equilibrium dialysis, which are used to study the binding constants between small molecule, big protein, two big proteins, and particularly, what are the limitations this, because in dialysis basically it is based upon the issue of the sizes. So equilibrium dialysis is limited to understanding or calculating the binding constant. But one partner has to be of relatively much larger size than the other partner, and then we can determine the binding constant between these two species by using these techniques. All this and more we will look at it during this module. So the concepts which are involved are that the antigen binding site almost always shows multi-specificity, that is, it is not absolutely specific. We will also learn that as bivalent immunoglobulin molecules create a network with multivalent antigen, avidity is far higher than simple binding constant between monovalent FAB and this antigen complex. The binding between a heptane and FAB largely depends upon close complementarity of the shapes of the two entities as shown in the diagram. This is due to inherent nature of the weak forces which are involved in the binding. We had earlier referred to the elegant studies of Wu and Kabat in identifying the hypervariable regions, and that gave the first clear clue about the nature of the antigen binding site. We had also referred to the affinity labeling experiments by Singer, which confirm the role of these regions in binding site of the antigen. Now that the fine structure of the antibody molecules are available, and we see that the antibody molecule consists of various domains. Each domain is characterized by two sheets of antiparallel beta strands joined by a SS bond. This kind of a structural motif was first observed in antibodies. So this in fact is now called the immunoglobulin fold, and it has been found to occur in few other classes of proteins as well. The divergent evolution of a duplicate genes probably led to one evolving as the constant region and another as the variable region. It is believed that the evolutionary changes in the amino acid sequence led to the concave shape of the antigen combining site. The shape of the site exposes a large surface area to the antigen, and this allows a close fit between the antigen and the combining site. This stereochemical fit allows these two molecules to have multiple non-covalent interactions. X-ray studies have given us lot of insight about the nature of the antigen binding site. As an early example, X-ray studies revealed the binding of the heptane phosphorylcholine to the wedge-shaped cavity of the FAB fragment of a mouse immunoglobulin. The choline group of the heptane interacts with the hypervariable loops from both heavy and light chains. Its positively charged nitrogen is in a position to interact with the negative charge on the glutamate side chain. These X-ray studies indicated that the antigen combining site consists of loops of hypervariable sequences fixed to more rigidly constructed other parts of the variable domain which have more of a framework function. It is interesting to look at the similarity between antigen binding site of the antibodies and the active site of enzymes. In the case of the active site of the enzymes, the classical studies on lysozyme by John Ruppel had indicated that hexaoligosaccharide of N-acetylglucosamine fits well with the binding site of the enzyme. We had also referred to the antibody raised against dextran and looking at its binding constants with oligomers of the glucose. You will recall that those studies also revealed that the immunoglobulin binding site had space for about 6 glucose residues. It works out to be about a length of nearly 25 angstrom. Another feature which we can compare is the microenvironment of the binding sites of the enzymes and the antibodies. Densyl lysine is an interesting heptane which has both hydrophilic and hydrophobic component. The fluorescence emission spectra of the free heptane compared with its spectra when bound to its antibody showed decrease in the lambda max of emission and significant increase in the fluorescence intensity. Both of these changes in the fluorescence spectra indicate that the microenvironment of this heptane binding site is predominantly nonpolar. It is known from the structure of the various enzymes that the active site of the enzymes also have predominantly hydrophobic character. Thus, the microenvironment of the binding sites of the two biological molecules, antibodies and enzymes seem to be designed in a fairly similar way. However, there is a fundamental difference between the two classes of proteins. In the case of enzymes, binding of the substrate leads to a catalysis. In case of antibodies, heptanes or antigens are not chemically transformed. We will of course may later on talk briefly, or you will read about it elsewhere, about the catalytic antibodies, which takes this analogy between the two classes of proteins further. In this respect, antibodies are closer to lectins, which specifically bind to sugars or the glycoconjugates. The biochemistry of lectin is a relatively new area as compared to the immunoglobulins and antibodies and immunology. But it is interesting to note that the early work on the lectins was pioneered by the same group of scientists who later on, who were earlier working with immunoglobulins and also started working with the lectins. We have so far focused on how the recognition of an antigen by antibody becomes specific. How do we reconcile this with cross reactivity of antibodies? How does that occur? Let us look at this diagram. What we can see is that a single antibody can in fact bind to more than one antigen, and here we are not referring to many antigenic determinants on the same antigen. Antibodies can bind to the antigenic determinants which was used to raise the antibody. It can also bind to the antigenic determinants with similar structures. This is referred to as cross-reacting antigens. Antigenic determinants with very different structures can also bind to an antibody. A stable antigen-antibody complex can form if there are enough short-range interactions. The nature and number of these short-range interactions may be different from what are involved in the recognition of a specific antigenic determinant. This phenomenon is called multi-specificity of antibodies, and in fact also contribute to the diversity of the antigen binding capacity of the immune system. Let us try to understand this phenomenon of multi-specificity of antibodies. Dan Taufik's group at the Weizmann Institute of Sciences in Israel has been looking at instances of multi-specificity shown by some antibodies. Taufik's group incidentally also studies the phenomenon of catalytic promiscuity by enzymes. We have earlier discussed the similarity between enzyme substrate and antigen-antibody recognition phenomenon. Catalytic promiscuity in a way refers to the multi-specificity of the enzymes. For example, a hydrolase like lipase is now known to synthesize carbon-carbon bond in reactions like aldol reactions. This phenomenon has been discussed more in the modules related to the lipids, as lipases are extensively studied class of enzymes vis-a-vis catalytic promiscuity. So in a way, this multi-specificity by both antibodies and enzymes again realize that the design of their binding site, which initiates the molecular recognition process, is very similar. Taufik's group used X-ray crystallography and pre-study kinetics to look at the phenomenon of multi-specificity of antibodies. Their studies have led them to the conclusion that one of the conformational isomer, which is a part of several conformations of antibodies in equilibrium with each other, can possess a low affinity promiscuous binding site for many heptanes. It is the subsequent induced fit which leads to the high affinity complex formation. In the latter, antigen binding sites assumes a narrower and deeper shape. Let us look at the binding constants of the antibodies. Binding with an unacceptable ligand leads to its dissociation due to global or local conformational changes, whereas specific ligands lead to lock-in conformational isomer. The binding sites for a heptane are deep, narrow cavities. Antigens, on the other hand, bind to a long and flat shaped surface of the binding site. Thus, antibodies have binding capabilities to a very wide range of antigens which differ in size and shapes. It has been already pointed out that there is a great similarity between the recognition of an antigen by an antibody and enzyme substrate interactions. In the latter case, in many cases, it is now well established that subsequently covalent enzyme substrate intermediates are formed. In the case of antigen and antibody, the binding forces, however, are purely non-covalent. These are similar to those which operate in many specific biomolecular recognition phenomena. Electrostatic forces, for example, is an important part of this. Many of these non-specific interactions require an optimum distance between the two entities for starting to operate. The nature of the forces involved in the antigen-antibody bindings are thus hydrogen bonds, electrostatic forces, van der Waals forces, or hydrophobic interaction. The dielectric constant of water is quite high with a value of 80. In vacuum, its value is 1. Hence, exclusion of water molecules between the two interacting charged surfaces will greatly enhance the force of binding. In addition to charges, dipoles on the antigen and antibody can also contribute to such forces. Charge transfer complexes, because of the electron donating groups on the antibody and electron accepting groups on the antigen, such as dinitrophenol, can also be formed. Because of the nature of the electrostatic interactions, the binding between antigen and antibody becomes large if the fit is close and shapes are complementary. The structure of the contact regions between a monoclonal FAB of the anti-lysozyme and lysozyme are shown in the figure. A space filling model shows that the FAB and lysozyme molecules fitting together smugly to form the complex. FAB and lysozyme models pulled apart in the B figure to show how the protuberances and depressions of each are complementary to each other. Hydrogen bonds can form between charged as well as uncharged species. The one to which hydrogen atom is originally attached is called hydrogen donor. The species which starts sharing this hydrogen atom is called hydrogen acceptor. Typically, N and O atoms bound to H in biological systems act as H donors. Similarly, atom on the acceptor species is also nitrogen and oxygen. The bond energies of hydrogen bonds are in the range of 3 to 7 kilocalories per mole. That is why hydrogen bonds are included in the list of weak bonds. While weaker than covalent bonds, hydrogen bonds are stronger than van der Waals bonds. Hydrogen bonds are directional in nature. Thus, hydrogen bond is strongest when donor of the hydrogen atom, and acceptor are collinear. While the affinity or affinity constant is well understood, the term avidity has often been a source of confusion. The familiar analogy is from the coordinate chemistry, where chelates have a high binding constants. So the term avidity is used to express the binding between multivalent antigen and antibody. It is applicable even when antisera is used as an antibody preparation, which actually consists of a population of antibodies, each binding to antigen or antigenic determinant with a different binding constant. It also includes the binding between antibody molecule to different but similar antigenic determinants which are present on the antigen. Van der Waals forces are non-specific attractive forces which start operating where two atoms are about 3 to 4 angstrom apart. These forces arise from the fact that at this distance, transient asymmetry of the charge around an atom induces similar distribution of charges. The bond energy of a van der Waals bond is about 1 kilocalories per mole. How weak is that can be appreciated from the fact that at room temperature, the average thermal energy of a molecule is about 0.6 kilocalories per mole. A significant contribution of these forces to the overall interactions between antigen and antibody arises from complementarity of the surfaces between the two that allows multiple van der Waals bonds to form simultaneously between the antigen and the antibody. Water is a polar molecule, and water molecules can form hydrogen bonds with each other. In water, these hydrogen bonded structures are continuously forming and breaking apart. Statistically, there is some order in the structure of water. More important, it arises from the fact that the strength of the multi-links is more than the sum of the strengths of individual links. Heterogeneity of IgG antiheptane, the heptane being the dinitrophenyl group. These antibodies from the serum of an immunized animal contrasting with the homogeneity of a monoclonal IgG anti-DNP is shown. The A part of the figure shows a sketched plot of the heptane binding to antibodies purified from the serum. The B part is a histogram which shows a typical distribution of antibody affinities in the anti-DNP serum. This figure further explains the bonus effect of multivalent attachment on binding strength, that is, the mechanism by which the avidity is more than the sum of its component numbers. Let us look at how the free energies are related in the cases the affinity and avidity. Let us assume that the individual links have K1 and K2 as affinity constants. In that case, delta G1 equal to minus RT ln K1 and delta G2 is equal to minus RT ln K2. Delta G avidity in that case is the sum of the delta G1 and delta G2, which ultimately becomes it is minus RT ln K1 into K2, which actually represents the minus RT ln K of avidity. Polyvalent interactions are actually very common in biological systems. Avidity refers to the association constant of a polyvalent interactions. Equilibrium dialysis can be used for calculating the binding constants. To start with, the antibody is placed in the dialysis sac. The heptane can diffuse into the bag. The heptane can be either measured spectroscopically or labeled with a radioisotope. The reversibility of the process can be checked by continually changing the buffer in which the heptane was dissolved in the vessel, that which is outside the sac. Measurement of the total heptane concentration in the dialysis bag enables the amount bound to the antibody to be calculated. The data can be plotted in terms of sketched plot as is shown. This plot also allows one to calculate the number of binding sites in the antibody molecule. At infinite concentration, R by C equal to 0, at that point the equilibrium reduces to Kn equal to Kr, hence the intercept on the x-axis equals N, which is the number of binding site. If all the antibody molecules in the preparation are identical as in the case of a monoclonal antibody, the slope of the sketched plot gives the association constant K, otherwise the plot will give the average association constant. In the case of a heptane, the mixture of antibodies produced by raising antisera against heptane carrier conjugate will in fact give number of binding sites less than 2, as many will not bind to the heptane at all. At a certain value of C, which is the concentration of free heptane, half of the antibody sites are occupied. At that point, the concentration of the antibody heptane carrier is equal to the concentration of antibody. Thus, K, the binding constant, is defined as the affinity constant and is equal to the heptane concentration when half the binding site of the antibodies are occupied. So students, let's now quickly summarize what we have learnt in this module. We have learned that the binding of a heptane with an antigen binding site involves many weak forces. The binding of course depends upon their synergy. Antibodies can cross-react and show multi-specificity. It is necessary to think in terms of avidity rather than simple affinity constant while discussing these antigen-antibody bindings. Many other methods for determining binding constants between antigen and antibody are available. These are like the methods which are used in the other cases, the interaction with the protein molecules. These include gel filtration, spectroscopic techniques. A technique which has proved very useful in calculating on rates and off rates is Biot. We will discuss it briefly in the context of its applications, which will come up in the later lectures. Thank you.